Good morning. Today we are going to start our next topic, somatic embryogenesis and artificial seed. In this topic, we will see how somatic cells can be used for the production of embryos. And in nature, we will see these embryos can able to produce from the genetic cells. But in artificial environment or in vitro condition, we can able to produce these embryos from the somatic cells. And we can utilize these embryos after the production to produce the artificial seeds, and artificial seeds we can use in the future, maybe 6 months to 1 year. We can preserve it and then we can utilize for the production of the healthy plants. So we will start with the somatic embryogenesis, then we will go for the artificial seeds. First, we will see the somatic embryogenesis. So the development of adventitious organ or primordia from undifferentiated cell mass in tissue culture or by the process of differentiation is called organogenesis. When adventitious organ or primordia is differentiating from the somatic cells, so that we are calling the as an organ cells. So in this topic, we will learn that how the somatic cells can able to produce the embryos. So it produce either root organ or shoot organ, and various vascular bundles also will be generated when we are culturing these root organ and shoot organ. So somatic embryogenesis is a process by which somatic cells or tissues, including haploid cells, can able to develop into the differentiated embryos, and they can able to regenerate into the entire plantlets. So in the laboratory condition, we can able to produce these embryos from the any type of cells. Since we know that a plant cells is a totipotent, so that it can able to produce a somatic embryos, and we can utilize to produce those plants which can't able to generate the embryos in nature. So those type of plant which can we can be able to produce the embryo. So if you see the history, Stewart and their colleagues in 1958, they first introduced the embryo through the suspension culture in the carrot, and they started the embryo production in the callus tissues. Then Greenard in 1959, he produced the embryo from the callus in carrot through the suspension culture. So in the suspension culture of the carrot cells, the Greenard produced the embryo from the callus. So from the callus and directly from the cells, Stewart and Greenard is the pioneer in the somatic embryogenesis. Now, importance of somatic embryogenesis, we'll see the higher propagation rate it is there in when we are culturing the somatic embryogenesis. When our target is to produce high number of the plantlets, so that we can utilize the somatic cell to produce embryos, and these embryos can able to utilize for higher number of plant production. Then it is also suitable for in the suspension culture. So when we want to increase the number of cells, so we can utilize the suspension culture, and using suspension culture, the number of cells can be increased, and this increased number of cells can be utilized to produce more somatic embryos, and later on we can utilize for the higher propagation of the plantlets. And artificial seed production also is another area where we can utilize these somatic embryos for the production of artificial seeds, and later on these artificial seeds can able to utilize for the production of the plantlets. Then labor saving, since we when we are producing the embryos, somatic embryos, so these embryos can be utilized to produce the plantlet directly. So it is a labor saving techniques, and these labor saving techniques can be utilized for the production of many plantlets. Now stages of somatic embryogenesis. So here we have somatic cells, and these somatic cell when we are culture on the artificial media and aseptic condition, this somatic cell can able to divide, and using mitosis it will divide into the vasculate cells, and these vasculate cells can able to induce to produce the somatic embryo by the phytohormone concentration, and different species we have to use the different concentration of the phytohormone. So one cell can able to convert into two cell protein, and this two cells can able to convert into the quadrant stage. This quadrant stage we can able to see in the microscope, and it will be how we are having this photograph. It will be look like in the microscope. Then this quadrant stage can able to convert into the octant stage. From the octant stage, we will get the dermatogen stage. Dermatogen stage can able to transform into the early globular stage, and early globular stage we can able to visualize by the naked eye, and this early globular stage can able to transform into the mid globular stage, and then we are giving the cytokinin. It will transform into the transition stage, and then it will convert into the heart shaped stage. Heart shaped stage can able to convert into the linear cotyledon stage, and these cotyledon can have the very specific three parts: apical part, and central part, and basal part. And apical part can able to produce the shoot, and basal part can able to produce the fruit. So these different stages is there in the somatic embryogenesis, and from single cell we can able to generate a somatic embryos, and these cells can be able to visualize or characterize when we are culturing and producing somatic embryogenesis to single cell. Now, type of somatic embryogenesis. So there are two type of somatic embryogenesis. First one, direct somatic embryogenesis, and second one is indirect somatic embryogenesis. When we are producing the somatic embryos from the direct somatic cells without producing the callus formation, then we are calling the direct somatic embryogenesis. Embryos are formed due to pre-embryonic determined cells, and without converting to the callus, we can able to convert the somatic cell into the embryos. And this method we are calling direct and somatic embryogenesis. Since we are not producing the callus directly, we are producing the embryos from the somatic cells, so that we are calling somatic embryogenesis. The next type is indirect somatic embryogenesis. An indirect method we are calling, since in between we are producing the callus from the somatic cells, and these callus can able to utilize to produce the embryos. So from callus, from cells, for somatic cells we are producing callus, and from callus we are producing the somatic embryos. That's why we are calling indirect somatic embryogenesis. So embryos are formed due to the induced embryogenic determinant cells. That's why we are calling embryos from the callus. And the reason behind that we are calling induced embryogenic determinant cell, because we are giving phytohormone concentration, and we are inducing these cells to produce embryos from the somatic cells. So here are the some research, here are the some experiment where the Indian meat genotypes we are utilized to produce somatic embryos, and direct somatic embryogenesis is there, and this direct somatic embryogenesis you can able to see these somatic cells is there in the petri plates, and through these somatic cells we generated. So in the direct and somatic embryogenesis, here you can see in the petri plate many colonies is there of the somatic cells, and these somatic cells can able to induce to produce the somatic cells. In this photograph you can able to see the somatic cells, and these somatic cells can able to utilize for the production of the plant head, and this wheat variety can able to produce from this direct somatic embryogenesis. Then here is the indirect somatic embryogenesis. Here is the indirect somatic embryogenesis. Here in the photograph you can able to see, first we produce the callus from the somatic cells, and here is the callus photograph, and from the callus we induce the embryogenesis, and these embryos can be utilized for the production of the plant roots. And here these callus is produced from the male sex plant. Now, importance of indirect somatic embryogenesis. So indirect somatic embryogenesis has high plant regeneration capacity. That's why we can able to produce more and more number of plantlets from the embryos, and these somatic embryos can able to regenerate very effectively. That's why number of plant generation will be high. Then indirect somatic embryogenesis reduce the breeding cycle, and we can able to breed very easily, and these embryos can able to utilize to reduce the breeding cycle. Then indirect somatic embryogenesis are also used in the crop improvement and improve variety we can able to produce through the somatic embryogenesis. And frequency of somatological variation is also very high in the indirect somatic embryogenesis, since we are again and again subculturing so that genetic material of these somatic clones can be induced artificially, and the somatic variation can be induced so that the plant varieties have new type of characters, and these characters can be utilized to select different new type of varieties, and these new type of varieties can be able to utilize to produce the somatic embryos, and these somatic embryos can able to produce the planters, and those planters after the hardening we can supply in the market or farmers. Then next, indirect somatic embryogenesis are better than the direct somatic embryogenesis, since indirect matter can be utilized to produce many type of can able to utilize to produce. Then indirect somatic embryogenesis are more useful compared to direct somatic embryogenesis, since this indirect method we are producing the callus. Many experiment on the callus, and through callus also we can improve the varieties of the somatic varieties of the crop, so that we can it will be useful for the crop improvement. And somatonormal variation when we are doing, so somatic indirect method of the somatic embryogenesis is more useful in this somatonormal variation compared to direct somatic embryogenesis. If you will see the somatic embryogenesis in monocot and dicot, they are totally different. Monocot radical protected by the coloriza and plumale polyptiles in monocot is there. If you will see the dicot, polyptiles and coloriza are totally absent. If you will see the monocot, cambial tissue are absent in monocot, whereas the in dicot cambial tissue are present. In monocot, secondary growth are absent, and in dicot, secondary growth are present. Then if you see the monocot somatic embryos easy to culture in the media, and dicot it is very difficult to grow in the artificial media. Next topic is artificial seeds. Many plant varieties they are very difficult to multiply by conventional propagation methods, and we want to improve through the traditional breeding methods. And for this purpose, we are producing artificial seeds, and for this purpose we are producing artificial seeds. So among the innovative technique for micropropagation, the concept of somatic embryogenesis with synthetic seed production or artificial seed technology is very promising. So these cultured embryo from somatic embryos we can utilize to produce the artificial seeds. Synthetic seeds or artificial seeds or lab produced seeds, they are all same, and is referred to as encapsulated somatic embryos. So when we will encapsulate the somatic embryos, we are calling the synthetic seed or artificial seeds, which functionally mimic the seeds and can develop into the seedling under the suitable laboratory condition. So these artificial seeds we can able to utilize to produce the plantlets in the laboratory condition. And what we have to do whenever we have demand in the market, so these seeds artificially synthesize somatic embryos or encapsulated somatic embryos, we can take it out from the storage, and then we can put these seeds in the suitable condition or suitable media, so that they can germinate and they can able to propagate as a how these seeds is developing a plant from the in the natural condition. Likewise, and we can able to develop these synthetic seeds as the synthetic seeds in the plant plate. So research also has been focused on the use of vegetative propagals like auxiliary but adventitious workers, shoot tips, cormels, bulb, and protocoms for production of synthetic seeds. Various type of cells we can utilize to produce these synthetic seeds. In general, the seed propagated outside the hybrid can be multiplied through the tissue culture and propagated by using synthetic seed rapidly. And we can rapidly produce the plantlet from these somatic embryos or encapsulated somatic embryos. And if we want to store, we can store it, and later on we can. Next topic is artificial seeds. Many plant varieties, they are very difficult to multiply by conventional propagation methods. And we want to improve through the traditional breeding methods. And for this purpose, we are producing artificial seeds. And for this purpose, we are producing artificial seeds. So among the innovative technique for micropropagation, the concept of somatic embryogenesis with synthetic seed production or artificial seed technology is very promising. So these cultured embryo from somatic embryos we can utilize to produce the artificial seeds. Synthetic seeds or artificial seeds or lab produced seeds, they are all same and is referred to as encapsulated somatic embryos. So when we will encapsulate the somatic embryos, we are calling the synthetic seed or artificial seeds, which functionally mimic the seeds and can develop into the seedling under the suitable laboratory condition. So these artificial seeds we can able to utilize to produce the uh, plantlets in the laboratory condition. And what we have to do whenever we have demand in the market, so these seeds artificially synthesize somatic embryos or encapsulated somatic embryos, we can take it out from the storage and then we can put on these seeds in the suitable condition or suitable media so that they can germinate and they can able to propagate as a how these seeds is developing a plant from the in the natural condition. Likewise, and we can able to develop these synthetic seeds as the synthetic seeds in the plant plate. So research also has been focused on the use of vegetative propagals like auxiliary but adventitious workers, shoot tips, cormels, bulb, and protocoms for production of synthetic seeds. Various type of cells we can utilize to produce these synthetic seeds. In general, the seed propagated outside the hybrid can be multiplied through the tissue culture and propagated by using synthetic seed rapidly. And we can rapidly produce the plantlet from these somatic embryos or encapsulated somatic embryos. And if we want to store, we can store it, and later on we can go the function. Then if you see the monocot somatic embryos easy to culture in the media, and dicot it is very difficult to grow in the artificial media. Next topic is artificial seeds. Many plant varieties, they are very difficult to multiply by conventional propagation methods. And we want to improve through the traditional breeding methods. And for this purpose, we are producing artificial seeds. And for this purpose, we are producing artificial seeds. So among the innovative technique for micropropagation, the concept of somatic embryogenesis with synthetic seed production or artificial seed technology is very promising. So these cultured embryo from somatic embryos we can utilize to produce the artificial seeds. Synthetic seeds or artificial seeds or lab produced seeds, they are all same and is referred to as encapsulated somatic embryos. So when we will encapsulate the somatic embryos, we are calling the synthetic seed or artificial seeds, which functionally mimic the seeds and can develop into the seedling under the suitable laboratory condition. So these artificial seeds we can able to utilize to produce the uh, plantlets in the laboratory condition. And what we have to do whenever we have demand in the market, so these seeds artificially synthesize somatic embryos or encapsulated somatic embryos. We can take it out from the storage, and then we can put these seeds in the suitable condition or suitable media, so that they can germinate and they can able to propagate as a how these seeds is developing a plant from the natural condition. Likewise, same we can able to develop these synthetic seeds as a the synthetic seeds in the plant plate. So research also has been focused on the use of vegetative propagants like auxiliary but advantageous workers, shoot tips, formels, bulb, and protocoms for production of synthetic seeds. Various type of cells we can utilize to produce these synthetic seeds. In general, the seed propagated outside the hybrid can be multiplied through the tissue culture and propagated by using synthetic seed rapidly. And we can rapidly produce the plantlet from these somatic embryos or encapsulated somatic embryos. And if we want to store, we can store it, and later on we can utilize to produce these. Now here you can see in the photograph many artificial synthetic seeds is there, and in the encapsulation you can able to see embryos, somatic embryos is there, and these somatic embryos later on if you will cultured in the artificial media, they can able to produce the plantlets. Now advantages. First advantage is direct delivery of tissue cultured plant to the field. So when we are having these encapsulated somatic embryos, these somatic embryos we can directly put on the media. Now first advantage is direct delivery of tissue culture plants to the field. When we are culturing the encapsulated somatic embryos, so that we have to put these somatic embryos on the artificially synthesized media and in vitro condition, so that these encapsulated somatic embryos can able to generate plantlets, and after the hardening we can able to directly transfer into the field. So n number of plant it will be very easy to produce from these synthetic seeds. Then propagation of desirable genotype with the genetic uniformity we can able to produce. We know that the somatic embryos is the clones of the single cells, and these single cell clone produce this single cell can able to produce n number of embryos, and these encapsulated embryos can able to have same genotype and genetic uniformity. That's why when we are growing the plant from the synthetic seeds, these plants have the uniform genetic types, and the experiment if you want to perform, then you can easily perform the same type of genotype material. Then reduction in cost of vegetative propagated elite plants. When we are propagating elite type of plants, so it is very hard to propagate vegetatively these elite type of plants, since they are having some natural barrier. So when we are propagating using the somatic embryos, and when we are culturing these somatic embryos, so they can able to produce the n number of plantlets. So we can store using the synthetic seed, and whenever we are having requirement, we can able to propagate how much the number of required elite plant we have. Then preservation of germ plants and convenience in germ plants exchange. So when we are propagating somatic embryos, so these somatic embryos we can able to preserve in the encapsulation, and these encapsulated somatic embryos we can able to generate the plantlets whenever we have requirement. Or otherwise, if you can able to preserve, you can preserve it in the suitable condition. And reduction in the dormancy period. Since these somatic embryos, whenever we want, we can able to produce the plantlets. So there is no dormancy periods, and according to market demand, we can able to produce whenever we have requirement of the production of the plantlets. And large production identical embryos in short time we can able to do. So we already know these somatic embryos is identical to the somatic cells, and from the single cell, since we are producing and their genotype is identical or cloned one, so that the identical embryos in very short time we can able to produce in the large quantity. Then it can be produced throughout the year, and it is not dependent. There is no dormancy, and suitable laboratory environment, laboratory condition if we will provide. So throughout the year we can able to produce these somatic embryos from the any type of plantlet. So synthetic seeds is basically a living seeds like structure derived from the somatic embryos, and inside the living tissues is somatic embryos, and the seed coats you can have to put these somatic embryo into the encapsulation. So seed coat work as a encapsulation, and your somatic cells is a living part which can able to propagate as a plant head. So encapsulation by a very various hydrogel you can able to do, and if you see the synthetic seeds. Synthetic seeds is there is two type of structure the synthetic seed, and the inner part or living part is the somatic embryo, and somatic embryo we have to encapsulate, and this encapsulation can able to compare with the seed coat, and these encapsulation we can able to produce by the hydrogel and other type of protective gels like calcium alginate, and these protective encapsulation it will provide a barrier against the microbes and desiccation, and since this hydrogel is containing the water, and so that it will be against the desiccation of the somatic embryos. Here in the flow diagram, you can able to understand how we can able to produce the somatic embryos, and then somatic embryos can be utilized to produce the synthetic cells. So here if we are having somatic cells, so somatic cells in the directly we can able to produce the somatic embryogenesis, or we can able to produce the callus, and these callus cells can be utilized to produce the somatic embryos. So embryos produced directly or indirectly can be utilized to produce the synthetic seeds. So isolated embryos mixed with the sodium alginate solution, and this alginate solution then we will take it out using the proper, and then we will put into the 30 to 100 millimolar calcium nitrate solution. So these calcium nitrate solution, when we will be in the contact of sodium alginate, it will produce the encapsulation. Then this encapsulated embryos we can take it out from the calcium nitrate solution, and we can keep it in the petri plate for the 6 to 8 months, and after this in between the 6 to 8 months if we have requirement we can put into the media, so that this plantlet these somatic embryos can able to induce and produce the plantlet. If you want to test it the how the somatic embryos having capacity to produce the plantlet, you can go to test it, and some 5 to 6 embryos randomly you can choose, and you can put on the artificial media. They can able to produce the plantlets. And if you want to produce the plantlet, you can directly use the vermicompost, and this sterilized vermicompost can able to germinate the plants, and you can harden these plantlets, and you can directly plant into the planting pot, or you can give into the farm country pot or ocean. Then if you see the monocot somatic embryos easy to culture in the media, and dicot it is very difficult to grow in the artificial media. Next topic is artificial seeds. Many plant varieties they are very difficult to multiply by conventional propagation methods, and we want to improve through the traditional breeding methods. And for this purpose, we are producing artificial seeds, and for this purpose we are producing artificial seeds. So among the innovative technique for micropropagation, the concept of somatic embryogenesis with synthetic seed production or artificial seed technology is very promising. So these these cultured embryo from somatic embryos we can utilize to produce the artificial seeds. Synthetic seeds or artificial seeds or lab produced seeds, they are all same and is referred to as encapsulated somatic embryos. So when we will encapsulate the somatic embryos, we are calling the synthetic seed or artificial seeds, which functionally mimic the seeds and can develop into the seedling under the suitable laboratory condition. So these artificial seeds we can able to utilize to produce the plantlets in the laboratory condition. And what we have to do whenever we have demand in the market, so these seeds artificially synthesize somatic embryos or encapsulated somatic embryos, we can take it out from the storage and then we can put these seeds in the suitable condition or suitable media so that they can germinate and they can able to propagate as a how these seeds is developing a plant from the natural condition. Likewise, and we can able to develop these synthetic seeds as a the synthetic seeds in the plant plate. So research also has been focused on the use of vegetative propagals like auxiliary but adventitious workers, shoot tips, formels, bulb, and protocoms for production of synthetic seeds. Various type of cells we can utilize to produce these synthetic seeds. In general, the seed propagated outside the hybrid can be multiplied through the tissue culture and propagated by using synthetic seed rapidly. And we can rapidly produce the plantlet from these somatic embryos or encapsulated somatic embryos. And if we want to store, we can store it, and later on we can utilize to produce these. Now here you can see in the photograph many artificial synthetic seeds is there, and in the encapsulation you can able to see embryos, somatic embryos is there, and these somatic embryos later on if you will cultured in the artificial media, they can able to produce the plantlets. Now advantages. First advantage is direct delivery of tissue culture plant to the field. So when we are having these encapsulated somatic embryos, these somatic embryos we can directly put on the media. Now first advantage is direct delivery of tissue culture plants to the field. When we are culturing the encapsulated somatic embryos, so that we have to put these somatic embryos on the artificially synthesized media and in vitro condition so that these encapsulated somatic embryos can able to generate plantlets, and after the hardening we can able to directly transfer into the field. So n number of plant it will be very easy to produce from these synthetic seeds. Then propagation of desirable genotype with the genetic uniformity we can able to produce. We know that the somatic embryos is the clones of the single cells, and these single cell clone produce this single cell can able to produce n number of embryos, and these encapsulated embryos can able to have same genotype and genetic uniformity. That's why when we are growing the plant from the synthetic seeds, these plants have the uniform genetic types, and the experiment if you want to perform, then you can easily perform the same type of genotype material. Then reduction in cost of vegetative propagated elite plants. When we are propagating elite type of plants, so it is very hard to propagate vegetatively these elite type of plants since they are having some natural barrier. So when we are propagating through, using the somatic embryos, and when we are culturing these somatic embryos, so they can able to produce the n number of plantlets. So we can store using the synthetic seed, and whenever we are having requirement, we can able to propagate how much the number of required elite plant we have. Then preservation of germ plants and convenience in germ plants exchange. So when we are propagating somatic embryos, so these somatic embryos we can able to preserve in the encapsulation, and these encapsulated somatic embryos we can able to generate the plantlet whenever we have requirement, or otherwise if you can able to preserve, you can preserve it in the suitable condition. And reduction in the dormancy period, since these somatic embryos whenever we want we can able to produce the plantlets, so there is no dormancy periods, and according to market demand we can able to produce whenever we have requirement of the production of the plantlets. And large production identical embryos in short time we can able to do. So we already know the somatic embryos is identical to the somatic cells, and from the single cells since we are producing, and their genotype is identical or cloned one, so that the identical embryos in very short time we can able to produce in the large quantity. Then it can be produced throughout the year, and it is not dependent. There is no dormancy, and suitable laboratory environment, laboratory condition if we will provide. So throughout the year we can able to produce these somatic embryos from the any type of plantlets. So synthetic seeds is basically a living seeds like structure derived from the somatic embryos, and inside the living tissues is somatic embryos, and the seed coats you can have to put these somatic embryo into the encapsulation. So seed coat work as a encapsulation, and your somatic cells is a living part which can able to propagate as a plant head. So encapsulation by a very various hydrogel you can able to do, and if we see the synthetic seeds, synthetic seeds is there is two type of structure in the synthetic seed, and the inner part or living part is the somatic embryo, and somatic embryo we have to encapsulate, and this encapsulation can able to compare with the seed coat, and these encapsulation we can able to produce by the hydrogel and other type of protective gels like calcium alginate, and these protective encapsulation it will provide a barrier against the microbes and desiccation, and since this hydrogel is containing the water, and so that it will be against the desiccation with the somatic embryos. Here in the flow diagram, you can able to understand how we can able to produce the somatic embryos, and then somatic embryos can be utilized to produce the synthetic cells. So here if we are having somatic cells, so somatic cells in the directly we can able to produce the somatic embryogenesis, or we can able to produce the callus, and these callus cells can be utilized to produce the somatic embryos. So embryos produced directly or indirectly can be utilized to produce the synthetic seeds. So isolated embryos mixed with the sodium alginate solution, and this alginate solution then we will take it out using the proper, and then we will put into the 30 to 100 millimolar calcium nitrate solution. So this calcium nitrate solution, when we will be in the contact of sodium alginate, it will produce the encapsulation. Then this encapsulated embryos we can take it out from the calcium nitrate solution, and we can keep it in the petri plate for the 6 to 8 months, and after this in between the 6 to 8 months if we have requirement we can put into the media, so that this plantlet these somatic embryos can able to induce and produce the plantlet. If you want to test it the how the somatic embryos having capacity to produce the plantlet, you can able to test it, and some 5 to 6 embryos randomly you can choose, and you can put on the artificial media, they can able to produce the plantlets. And if you want to produce the plantlet, you can directly use the vermicompost, and this sterilized vermicompost can able to germinate the plants, and you can utilize, you can harden these plantlets and you can directly plant into the planting pot, or you can give into the farmers. So here is the structure of the artificial seeds, and in the somatic seeds you can see the somatic embryos is there, and the somatic embryos is encapsulated with the hydrogel, and hydrogels is work as a seed coat, and it will protect against the bacteria, it will protect against the microorganism and the desiccation. So artificial embryo, artificial seeds have artificial endosperm, and this hydrogel can able to function both type. It will provide protection and it will also provide how endosperm is giving the nutrition to the somatic embryos. So these two type of work this hydrogel can able to perform. Now the various encapsulation media we are utilizing for the synthetic seed generation purpose, like sodium alginate is there, so zero point 0.5 to 5 weight by volume we are utilizing in the calcium salts complex agent, and concentration of the iron we can put 30 to 100 micromolar. Then sodium alginate with the gelatin also we can use in combination of gelatin. Sodium alginate 2% with the gelatin 5%. We can able to use these two combination for the encapsulation in the complex agent of calcium chloride, and concentration will be 30 to 100 micromolar. Then kerosene is another hydrogen solution we can able to produce, and 0.2 to 0.8% weight by volume we can take, and potassium chloride and ammonium chloride we can use as a complex agent, and the ultimate concentration of the iron will be 500 micromolar. Then locust bean gum is also another encapsulation media we can utilize, and zero point 0.4 to 1 percent weight by volume we can utilize in again the complex agent of potassium chloride and ammonium chloride. Then gelrite is also another media is there, another chemical, and this gelrite can be utilized for the encapsulation 0.25% weight by volume. In the among these, the sodium alginate is the most suitable type of gel forming agent, and sodium chloride with the combination of gelatin also utilized for the encapsulation. So here in the photograph you can able to see the artificial seeds produced by the encapsulation in 2.5% sodium alginate, and somatic embryos is encapsulated, and when we are putting the somatic embryos encapsulated, we can able to germinate these. So here in the photograph you can able to see the somatic embryos encapsulated in 2.5% sodium alginate, and when the somatic embryos we can put into the artificial media so that the somatic embryos can able to germinate, and the somatic embryo can able to break down this artificial encapsulation, and they can able to produce the root and shoot form. Now some important books for further study. First book is Plant Cell and Tissue Culture by Indra K. Vasil and Trevor A. Thorpe. Second book is Plant Cell and Tissue Culture, a tool in biotechnology, basics and application, principle and practice by Carl Harman Newman and Ashwani Kumar. Next book is Plant Cell and Tissue Culture Methods in Molecular Biology by Jeffrey W. Pollard and John M. Walker. Then next book is Plant Cell and Tissue Culture for the Production of Food Ingredients by Tom J. Fu and Gurmeet Singh. So these are all reference books if you want to study further you can utilize. Now some important question in the somatic embryogenesis and the artificial seed production. So first question is the first step in somatic hybridization is option A is identification of hybrid cell, option B is protoplast fusion, option C is protoplast isolation, option D is identification of hybrid cell. Then we have to select any one correct answer. The next question, hybrid cell is made by fusing two protoplast space. Option A is callus culture, option B is suspension culture, option C is gene cloning, option D is somatic hybridization. So you have to select any one correct answer. The next question, space are the used for protoplast fusion? Option A is chemical agent, option B is fusion, option C is vitamins, option D is plant hormone. So again you have to select the anyone correct answer. The next question, space is the physiogenic agent, A polyvinyl alcohol, B ethyl alcohol, C propanolol, D option is methanol. So again you have to select the anyone correct answer. Next question, the following are the method used in the hybrid cell selection except option A is cytometric method, option B is biochemical method, option C visual method, and option D is PCR. So anyone right answer you have to select. Then in the synthetic seeds, we have some important questions. So first question is synthetic seeds are option A is artificial synthesized seeds. Option B is somatic embryos encapsulated in somatic matrix and suitable matrix. Option C seed of plant modified genetically. Option D natural seeds. Again, you have to select any correct answer, correct option. Then somatic. Next question is somatic embryos are option A is identical with zygotic embryos and without seed course. Option B identical with zygotic embryos and with seed course. Option C non-identical with zygotic embryos and without seed course. Then option D non-identical with zygotic embryos and with seed course. Then next question, the preserved embryos are termed as option A is synthetic seeds, option B is semi-synthetic seeds, option C natural seeds, option D fermented seeds. So again you have to select the correct answer. Then next question, encapsulation is necessary to produce and protect synthetic seeds. The encapsulation is carried out by various types of hydrogen, which are option A is soluble in water, option B soluble in organic solvents, option C insoluble in water, option C, option D insoluble in organic solvents. So again, you have to select any correct answer. The next question, which of the following is not true about the synthetic seeds? Option A is can be stored for a year without the loss of valuables. Option B easy to handle. Option C can be directly sown in the soil like natural seeds. Option D is need hardening in the greenhouse. So these are all these and some important questions, and we will see you in the next class. Thank you.